Hello my dear students, today we will see the another lecture in this series of lectures in plant physiology. In our last lecture we have seen the diffusion, and I am very much hopeful that this lecture series will be helpful to the students of UG, PG and other students who are preparing for the different competitive examinations in the life sciences, even for the net set as well as the jams. Friends, myself Dr. PITAMBAR HUMANE, founder Botany for You and working as Associate Professor and Head Department of Botany, Dharampeth M, Deo Memorial Science College, Nagpur, Maharashtra, India. I welcome you all in this online platform for the students of Life Sciences Botany for You. If you are new or not yet subscribed our YouTube channel Botany for You, then kindly like, share and subscribe our YouTube channel Botany for You, so that you can get the alerts of different lectures uploaded on our channel. If you want to associate with us through Telegram group, then you can use the link given in the description, so that you can also get the advantage of the different PDF books as well as the advertisements, appointments from India and abroad in life sciences. So let's see today's topic, that is osmosis. As in yesterday's topic we have seen the diffusion, and in diffusion we have seen that the transport of solid particles from the region of higher concentration to the region of lower concentration. We have yesterday taken the example of the dhoop or the agarbatti. Even we are smelling the smell of the deo, then perfume. So all these are the basic examples of the diffusion. If we put the indigo in a bucket in a water, then also we are able to see the simple process of the diffusion. Even when we are preparing the tea or the coffee, in which there is a equal distribution of the sugar and even the tea and the coffee, that is also the examples of the diffusion. So here this is the simple process, that is diffusion from the region of higher concentration to the region of lower concentration. There is a transport of solid particles. We must remember that the solid particle in the diffusion, though there is a movement of the solvent also. But when we are talking about the osmosis today, so first of all we must know the difference in between the diffusion and osmosis. So first of all, when we are talking about the osmosis, you must remember that in a osmosis there is a transport of the solvent molecules and not the solid. This is one thing. And another thing is that this transport of the solvent molecules, that is carried out through the semi-permeable membrane. Now let's see the definition. The transport of solvent particles or the molecules from the region of lower concentration to the region of higher concentration. See the difference. Solid particles from the region of higher concentration to the lower concentration in the diffusion, while the solvent particles in the osmosis from the region of lower concentration to the region of higher concentration. But if you see the basic principle behind it, it is same, that after the complete process of the diffusion, there is a formation of equilibrium, that the concentration of that particular solvent becomes equally distributed all over the body. In the similar manner, when the solvent or the two solvents or the solution and the solvent, they are separated from each other by means of a semi-permeable membrane, again there is a equal distribution of the concentration on both the sides. This is also nothing but the equilibrium. So this process is carried out till it reach the equilibrium, so that the concentration on both the sides of the semi-permeable membrane become same. Now see here, in the first side you will find that there is a lower concentration, and to the right side here there is a higher concentration, and both are separated from each other by means of semi-permeable membrane. So from the region of lower concentration to the region of higher concentration, there is a transport of solvent molecules, and because of the transport of the solvent molecules, here there is a reduction in the amount of solvent in the left side and increase in the amount of the solvent to the right side. But if you see the concentration on both the sides, then you will find that here there is a formation of similar type of the concentration. So till it reach the equilibrium, there is a continuous process of the osmosis, and in this way the osmosis is used for the movement of water in the plant body. Though the plasma membrane is not truly semi-permeable membrane, because in plants the water when transported or move through the plasma membrane, it can also allow some of the solids, and hence it is known as selectively permeable or differentially permeable membrane. In this diagram, we can easily observe or carry out the process of the osmosis. Here the potato is used as a osmoscope to study the osmosis. See here, there is a formation of notch in the potato, and this notch is filled with the sugar solution, and the lower part of this potato is peeled off, as the outer cover of the potato is impermeable to the water. Later on, the outer side is filled with the water, that is the beaker. So what happened here? The outer side contain the water, and inside the notch or in the osmoscope there is a presence of the sugar solution, and because of that, here from the region of lower concentration to the higher concentration, here there is a movement of water, which is the solvent here. We know every living organism, the cell sap contain the solvent as a water. So it was filled with up to this level initially, and hence it is known as the initial level, and after some time the water from outside will start to enter inside this osmoscope because of the higher concentration inside the notch, which is filled with the sugar solution, and because of that this level of this sugar solution now increases, which is considered as a final level. This means that here the water enter inside the notch because of the presence of the sugar solution. So here there is a process of the osmosis, and we can carry out this experiment very easily in the laboratory to demonstrate the osmosis with the simple process. This is another experiment, that is the demonstration of the osmosis with the help of thesulfonil. So here the thesulfonil is taken again, which is filled with the sugar solution. This is again exactly similar to that of the osmoscope. Instead of the osmoscope, we have taken here the thesulfonil. So initial level, this is the initial level of the sugar solution, and At the bottom, here the T cell funnel is tied with the semi-permeable membrane, that is by means of either goat bladder or even the egg membrane. And the outer side of the T cell funnel is filled with the water. So because of this, here the inside it contain the higher concentration and outside will have the lower concentration, and hence from the region of lower concentration to the region of higher concentration there is a movement of the water through the semi-permeable membrane, and hence here there is a presence of the semi-permeable membrane, and hence through semi-permeable membrane the water will enter inside the T cell funnel, and because of that here the initial level will now rise up and it is said to be the final level, and this increase in the level in the T cell funnel indicates that there is a entry of the water through the semi-permeable membrane from the region of lower concentration to the region of higher concentration, so that we can prove that there is a process of the osmosis, means the movement of the water from the region of lower concentration to the region of higher concentration through the semi-permeable membrane. Then again, on the basis of the entry of the water inside the cell and outside the cell, there are the different type of the osmosis, and that can be studied very well by keeping or putting the different type of the cells in a solutions of the different concentrations. Here we have the different solutions, that is the A, which is the isotonic solution, B, which is the hypertonic solution, and C, which is the hypotonic solution. Means here these beakers are filled with the isotonic solution, hypertonic solution and the hypotonic solutions respectively. Inside which there is a presence of balloon which is made up of the semi-permeable membrane. So we can say that if it is made up of the semi-permeable membrane, then the water can enter inside only and not the solids. Even instead of this balloon, we can take the cells also. So when it is kept inside the isotonic solution means here the concentration inside this balloon and the concentration outside the balloon is same. If there will be the similar type of the concentration, there will not be the any effect, because even may the water enter inside, but at the same time it can come outside also, because they have the same type of the concentration, or there will not be the entry of the water inside or not from inside to the outside. So there will not be the any change in such type of the cases when the cells are kept in a isotonic solution. But when the cells are kept in a hypertonic solution, it means that the inside the cell or the balloon is the lower concentration and outside is a higher concentration. And earlier we have seen from the lower concentration to the higher concentration there is a movement of the solvent molecules. So in this way here there will be the release of water, or the water will come outside the cell, and hence we can consider that this is X-osmosis, because the osmosis or the osmotic diffusion of the water is carried out outside the cell, and hence when the cell is kept in a hypertonic solution there will be the X-osmosis, that we should remember. In another case, when the cell or the balloon is kept in a hypotonic solution, hypotonic means here the outside is with the lower concentration and the inside sap is with the higher concentration, and as we know there is a entry or the transport of the solvent molecules from the region of lower concentration to the region of higher concentration. So the water will enter inside the balloon or the cell, and in this way here there will be the end osmosis. and because of the end osmosis, the cell or the balloon will be turgid. So here these will be the two types. So here when it will be kept in a hypotonic solution, the cell or the balloon will turgid, while when it is kept in a hypertonic solution, the cell or the balloon will be flaccid. So if the cell is kept in such a type of the hypertonic solution, there will be a plasmolysis. So we will see the plasmolysis separately in the different lecture. Now whenever we are talking about the plant cell, that is a osmotic system, because when we are talking about the osmosis, there is a transport of the solvent particles, and this transport of the solvent particle is because of the pressure, and this pressure is known as the osmotic pressure. Means now say for example, if there is a cell A and the another cell that is a B. Suppose now the cell A having the higher concentration and the cell B having the lower concentration, so that the water will start to move from the lower concentration to the higher concentration. It means that the osmotic pressure of the cell A is high and the osmotic pressure of the cell B is low. And if we talk about the osmotic pressure, then the osmotic pressure is directly proportional to the amount of solids dissolved in it. It means that the osmotic pressure is directly proportional to the concentration of the solution, and hence if the osmotic pressure will be higher, it will absorb the water from surrounding cells. So here in the cell, when we are talking about that the cell is kept in a hypotonic solution, it means that the water will enter inside, because when outside there is a hypotonic solution, means the solution with the less concentration, it will have the lower osmotic pressure, and the cell will have the higher osmotic pressure. So the water will move from the lower osmotic pressure to the higher osmotic pressure. But the cell having the cell wall and even the plasma membrane in the plant cell, cell wall is permeable to the water. Now if the water from outside is continuously entered because of the osmotic pressure, if it is kept in a hypotonic solution, it means that because of the entry of the water inside the cell, here again there will be the creation of the pressure inside the cell. So the osmotic pressure is always there because of the dissolved solid in it, and the pressure which is created by the plasma membrane, that is known as the turgor pressure. When the cell turgid, it creates the pressure on the cell wall, and hence this pressure is known as the turgor pressure. But at the same time, when the cell membrane or the plasma membrane create the pressure on the cell wall, at the same time the cell wall also create the pressure on the plasma membrane, and this pressure which is created by the cell wall is known as the wall pressure. If cell is fully turgid, at that time we can say that the turgor pressure will be equal to the wall pressure. So all these factors will be useful in understanding the upcoming topics like the diffusion pressure deficit and the water potential also. Here only we are able to understand that if the cell having the higher osmotic pressure, then it can absorb the water, but only the osmotic pressure is not responsible for the absorption of the water by the cell. So that we will see in a subsequent lecture. Now, whenever we are talking about the determination of the osmotic pressure of the cell sap by the plasmolytic method, earlier we have seen when the cell is kept in a hypertonic solution, at that time the water from the cell come outside, that is the X osmosis is done. So, in such a situation, here if we have to prepare the different solutions, and in a different solutions or with the known concentration, and the cells are kept inside it. So, in such a situation, what will happen here, if the cell is with the same concentration, that is a isotonic solution, there will not be any effect on the cell. But if it is kept in hypertonic solution, then there will be the X osmosis. If it is kept in a hypotonic solution, then there will be the end osmosis. So, if the cell is kept in a slightly hypertonic solution, there will be a little separation of the protoplasm or the plasma membrane from the cell wall. So, that solution having the known concentration will be nearly equal to the osmotic pressure of the cell sap, and in this way we can roughly identify the osmotic pressure of the cell sap by a plasmolytic method. As we have seen, it is slightly hypertonic. It means that the osmotic pressure of the cell sap will be slightly lower, and with the help of standard tables, again we can correct the value that we are obtaining from this experiment, and easily in the laboratory can find out the osmotic pressure of the different cell saps. Now, why we are studying the osmosis in the plant physiology? Because whenever we are studying any topic in a particular subject, there is a importance, function, or the significance. So, we are talking about such a significance or the importance of the osmosis. Number one, that is the absorption of water by roots. So, when we are seeing the absorption of the water by the roots, we have seen that there is a presence of the root hair zone, and this root hair zone is responsible for the absorption of the water from the ground. So, first of all, when there is a lower concentration or the lower osmotic pressure in the soil and higher osmotic pressure in the root hair, at that time automatically by the osmotic diffusion there is a entry of the water from soil to the root. So, this is one of the most important thing in the absorption of the water. Then opening and closing of the stomata. If you see the stomatal structure, then there are the presence of the guard cells, and these guard cells, when they turgid, the stomata get open, and when the guard cells remain flaccid, the stomata becomes close. It means that here the entry of the water inside the guard cell, that is the end osmosis, and when there is a X osmosis to leave the water by the guard cell, there is a closure of the stomata. This means that here the osmosis is completely responsible for the opening and closing of the stomata. The third one, that is the cell to cell movement of the water. Say for example, as we have seen earlier, here the cell A, this is the cell B and cell C and the D. Say for example, now if this cell D absorb the water, earlier all A, B, C, D cell having the same osmotic pressure, but because of the absorption of the water by the cell D, here the osmotic pressure of the cell D will be reduced as compared to the osmotic pressure of the A, B and the C. And as the cell C is in association with the D, the water will start to move inside the cell. See, as its osmotic pressure is higher than the osmotic pressure of the D. In the similar manner, after absorption of the water by the C, again the osmotic pressure of the C will be lesser than the osmotic pressure of the B. And in this way, from one cell to another cell, the movement of the water will be carried out by means of the simple process of the osmosis. Then maintenance of form and the shape of the organs. We know the different structures of the leaf, petals, flowers, etc. is completely depends upon the type of the cell, shape of the cell and its turgidity. And this turgidity is completely maintained by means of the process of the osmosis. Then even that is related with the resistance of the plant to the drought and the frost also. Even in such type of the condition, the higher osmotic pressure is maintained so that the plant can resist to the drought and frost condition. And when there is a germination of the seed, at that time during the germination, the embryo germinates and to form the plumule and the radicle. This plumule later on develops into the stem and this radicle develops into the root. So this is carried out, means the embryo come outside the seed pod, and when the seeds are sown inside the soil, even that plumule come outside the soil also. So this complete process is because of the increase in number of cell, and this increase in the number of cell because of the osmosis, because osmosis is responsible to maintain the turgidity of the cell, and so that it is again helpful in the germination of the seeds also. So in this way, the osmosis is helpful in various processes which are carried out in the plants, and hence it is very much important. In upcoming classes, we will see that only the osmotic pressure is not responsible for the absorption of the water. Instead of that, the diffusion pressure deficit or the suction pressure is responsible for the absorption of the water. So this we will see in the upcoming next lectures. I am very much confident and hopeful that this lecture will be helpful to you in understanding the osmosis and its utilization in the plants. Friends, if you like this lecture and if it is useful to you, then kindly like, share and subscribe our YouTube channel Botany for You, and also press bell icon for the alerts of every lecture to be uploaded on different life sciences titles. And don't forget to join our Telegram group with the link given in the description. Thank you very much for listening the lecture completely. Thanks a lot once again.